All right, welcome back to the deep dive. Today we're going to be really getting into like the nitty-gritty of how things get slowed down or even completely stop in biology.
hmm
We all want things to work properly all the time, but sometimes you really need to just stop them in their tracks.
Right.
And I think a lot of people listening are probably curious about like the underlying mechanisms of that. So we're going to dive into enzyme inhibitors and gene repressors. You sent over some great stuff on these topics.
Yeah.
And I think the goal today should be like to really understand the main principles behind these systems and why they're so important. But, without getting too bogged down in all the little details.
Absolutely. Yeah, you really want to be able to see the forest for the trees, get the essence of these regulatory mechanisms without getting lost in the weeds of all the molecular details.
Exactly. So we're going to be looking at these scientific explanations of how these molecules work. And hopefully, we'll come out of this with a really solid understanding. So let's jump right in. Let's talk about enzyme inhibitors first. What exactly are they doing at like the molecular level?
So enzyme inhibitors are basically molecules that decrease the rate of enzyme catalyzed reactions.
Okay.
And what's really interesting is they can be a lot of different things. They can be metabolic intermediates, they can be end products, which is kind of a signal saying, hey, we've made enough of this, let's slow down.
Right.
Or they can even be external substances, things like drugs or toxins.
So it's like the body's natural way of saying like, whoa, 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 slow down, or even like something external, like a drug can come in and do the same thing.
Yeah, exactly. And this just highlights how finely tuned biological systems are. There are all these different ways to really make sure things are operating at the right levels.
That's so cool. So the first type of inhibition that we're going to talk about is competitive inhibition. So how does that actually work?
Well, imagine the inhibitor as like a molecular imposter.
Okay.
It actually resembles the enzyme's usual target, which is called the substrate, and it competes for the enzyme's active site. And that active site is that specific pocket where the reaction actually occurs.
So it's basically just competing for like the working spot on the enzyme.
Exactly.
So what effect does that actually have on the enzyme's activity if this inhibitor is like hanging out in the active site sometimes?
Right. So because that active site is sometimes occupied by the inhibitor, you need more substrate to reach the enzyme's maximum speed.
Okay.
You can kind of think of it like if you're trying to get the right key in a lock,
Oh, yeah.
sometimes the wrong key's in there,
Yeah.
so you need more tries to get the right key in. But if you flood the system with enough substrate, you can eventually outcompete the inhibitor,
Okay.
and the maximum reaction rate stays the same.
Got it.
A good example of this is malonate. It inhibits an enzyme called succinate dehydrogenase, which is involved in cellular respiration
Oh, wow. So that's actually happening in our bodies right now.
all the time.
That's wild. Okay, and then we also have non-competitive inhibition. How is that different from competitive inhibition?
So in non-competitive inhibition, the inhibitor doesn't actually go for the active site at all.
Oh, interesting.
Instead, it binds to a different location on the enzyme, and this is called an allosteric site.
Okay.
And this binding, it causes the enzyme to change its overall shape, which in turn reduces its ability to catalyze the reaction.
So it's almost like it's like bending the tool so it doesn't work as well, even if the intended target is still there.
Precisely.
Oh.
And because the enzyme's fundamental efficiency is reduced,
Hmm.
you can't overcome this by just adding more substrate.
Right.
The maximum reaction rate will decrease.
Okay.
A good example of this is heavy metals interfering with enzyme function. They often work in a non-competitive way.
Oh, wow. So that's why heavy metal poisoning is so bad.
Exactly. They can
Right.
really mess with a lot of important enzymes.
Wow, that's scary. Okay, and then the last type of inhibition that we're going to talk about is uncompetitive inhibition.
Right.
What is that?
So this type is a little less common. Uncompetitive inhibitors only bind to the enzyme after the substrate has already attached.
Oh, okay.
And this forms an inactive complex that can't proceed with the reaction.
So it's almost like trapping the enzyme and the substrate together in a non-productive state.
That's a good way to visualize it.
Yeah.
And this type of inhibition, it affects both how readily the enzyme binds the substrate and its maximum speed.
So it's kind of like the worst of both worlds.
In a way, yeah.
Okay, so those are the three main types of enzyme inhibition.
Right.
Now, the material that you sent also mentioned feedback inhibition.
Yes. And feedback inhibition is really a beautiful example of biological self-regulation.
Okay.
It's often a form of allosteric inhibition, where the end product of a metabolic pathway goes back and inhibits an enzyme early in that same pathway.
Oh, wow.
So it prevents the overproduction of a substance.
So it's like the cell knows how much it needs, and then it tells itself to stop making it once it's got enough.
Exactly. It's a way to maintain balance and prevent waste.
That's amazing. So that's how enzymes can be directly controlled. Now let's talk about gene repressors. How do those work to stop things from happening at the genetic level?
Okay, so gene repressors actually prevent the production of enzymes in the first place.
Got it.
They're regulatory proteins that decrease gene expression. And they typically do this by blocking the very first step, which is called transcription.
Okay.
Transcription is the reading of DNA to make RNA, which then leads to protein synthesis. This is really a fundamental way that cells control what proteins are available, including enzymes.
So it's almost like they're controlling the blueprints for the enzymes.
Exactly.
So how do they block this reading process? How does that actually work?
So a repressor protein, it binds to a specific DNA sequence near the start of a gene, and this is called the operator.
Okay.
And this binding is highly specific and it's also reversible. When the repressor is bound, it physically blocks RNA polymerase. RNA polymerase is the enzyme that's responsible for reading the DNA and starting transcription.
So it's kind of like a roadblock on the DNA?
Yeah, you can think of it that way. It prevents the machinery from getting to the gene and reading it.
That's so cool. And these repressors, they can be influenced by other molecules, right?
Yes, that's right.
Like inducers and co-repressors?
Exactly. So in some systems, a repressor is only active when it's bound to a co-repressor.
Okay.
And this co-repressor is often the end product of the pathway. So when you have high levels of the end product, it activates the repressor, and then the repressor shuts down the genes that are making it.
Oh, so it's like another feedback loop?
Exactly. It's another way to maintain that balance.
It's like the cell is really good at not wasting energy.
Right. Very efficient.
That's so cool. The tropoperon is a prime example of this, right?
Yes, the tropoperon is a classic example of repression by a co-repressor.
Okay, cool. And then what about inducers?
Okay, so in other systems, the repressor is actually active on its own, and it's blocking transcription. However, when a specific inducer molecule is present, and this is often the substrate that the enzymes would normally process, it binds to the repressor. And this binding changes the repressor's shape and actually inactivates it.
Oh, so it's like the inducer is taking the roadblock away.
Exactly. And that allows transcription to proceed. The lactoperon, which controls lactose metabolism, works this way.
Wow, that's amazing. So to recap, we have enzyme inhibitors, which directly affect the activity of existing enzymes.
Right.
And then we have gene repressors, which control whether those enzymes are even made in the first place.
Exactly. And these two types of control mechanisms acting at different levels, they're essential for the really intricate regulation of all biological processes.
That's so cool. So for those of you listening, consider this. Understanding these fundamental on and off switches at the molecular level is absolutely critical for developing new therapies.
Yeah.
Think about it. Drugs are designed to inhibit a specific enzyme that's involved in a disease, or even modulate the expression of a particular gene. It's really amazing stuff. And what other incredibly sophisticated control systems might exist in biology that we're only just beginning to understand? It's definitely something to think about.
Definitely.
Thanks for joining us on this deep dive. We'll see you next time.